on enzyme-linked receptors, or enzyme-linked receptor mechanisms. For a little overview of what I'm going to be talking about, in case you don't remember from cell biology, I'm going to touch lightly on cell surface receptors, and then I'm going to generally talk about enzyme-linked receptors, and then I'm going to go more specifically into the 6 classes of enzyme-linked receptors and talk a little bit about their mechanisms, functions, and some research that has been done with them. Cell surface receptors. In order for a cell to function, there are signal molecules that are either go through the plasma membrane or they cause pathways to signal processes in the cell so that functions can occur throughout the body. In order to do this, sometimes the signal molecules are too hydrophilic or they are too large to go through the plasma membrane. This is when cell surface receptors are necessary to carry out these actions in the cells. Cell surface receptors help with cell communication between its environment and other cells, and these different cells in the body depend on different cell surface receptors, which will react a certain way depending on the signal that is binding to these receptors. There are three major categories of cell surface receptors, and these are ion channel linked receptors, G-protein coupled receptors, and enzyme-linked receptors, which I'll be talking about. Enzyme-linked receptors, also known as enzyme-coupled receptors or catalytic receptors, were first discovered during studies of growth factors, and they are the second largest group of cell surface receptors. These enzyme-linked receptors are transmembrane proteins, and they have a ligand binding domain, which evokes specific responses by different ligands binding to the same receptor because of high affinity for specific ligands. As you can see, here's the ligand binding domain, and enzyme-linked receptors have 3 domains. Like I said, there's the ligand binding domain, and then there's the transmembrane domain, which is made of hydrophobic amino acids, which hold the receptor to the plasma membrane. And then there's the cytosolic domain, which either acts as an enzyme or it associates itself directly with an enzyme. The enzyme-linked receptors cross the lipid bilayer once, unlike G-protein coupled receptors, which cross the membrane 7 times. There are 6 classes of enzyme-linked receptors. These are the receptor tyrosine kinases, the tyrosine kinase-associated receptors, receptor-like tyrosine phosphatases, receptor serine threonine kinases, receptor guanylyl cyclases, and histidine kinase-associated receptors. The first enzyme-linked receptor I'll be talking about is the receptor tyrosine kinase, which is the largest class of enzyme-linked receptors, and it's made of a monomeric protein chain that crosses the membrane 1 time and combines with another similar protein to form a symmetrical dimer complex. These also require 2 signal molecules to bind to the dimer complex that we have for the receptor, and these signal molecules are oftentimes growth factors. When these signal molecules attach to the receptor or bind to the receptor, they cause autophosphorylation of tyrosine residues. Autophosphorylation is the process of which cross-phosphorylation onto multiple tyrosines occurs between the adjacent kinase domains. And this is shown here's a diagram of the mechanism of the receptor tyrosine kinases. And as you can see, here's the one monomeric protein and the other, and then when the signal dimer binds to it, they come together to form the dimer complex, and then they activate this kinase, which then autophosphorylation occurs, and then you get these phosphates, and then they go on to perform the necessary functions in the cell. Receptor tyrosine kinases are the largest of the enzyme-linked receptor families, and they have 20 subfamilies, and because the signal that binds to them is a growth factor, they help to regulate the cell proliferation and differentiation in the cell. Because they help to regulate the different proliferation and differentiation of the cell, they oftentimes contribute to diseases and cancers found in the body, and these are caused by mutations in somatic germlines. Several mechanisms cause these, such as the autocrine growth factor loops. This is when the receptor tyrosine kinase is overexpressed in the cell, or the ligand is overexpressed when binding to it. An example of a receptor tyrosine kinase is the epidermal growth factor receptor. This can either come in a homo or heterodimer form, depending on the ligand that attaches to it. And it has been found that this is overexpressed in triple-negative breast cancer. They have done research on the inhibition of the epidermal growth factor receptor, and they did this and compared it to the HER2, which is a human epidermal growth factor receptor that is also highly expressed in the triple-negative breast cancer, and over the years it's been used as a predictive marker when searching for other therapeutic options for cancer. So what they did in this research was they took 3 inhibitors, gefitinib, erlotinib, and cetuximab, and which have been approved for treatment of a number of other solid tumors. And so what they found was that they didn't really affect, these inhibitors didn't really affect the way they didn't really suppress the epidermal growth factor receptors a whole lot, but, or as much as they did with the HER2, which they compared it to. And they also, what they did found was that the gefitinib inhibitor affected it more than the other 3. And they also found that when chemotherapy and the gefitinib worked together, that they were able to suppress the epidermal growth factor receptors better. Another enzyme-linked receptor is the tyrosine kinase-associated receptor. This is similar to the receptor tyrosine kinases, except that it lacks a tyrosine kinase domain. Instead, it works through cytoplasmic tyrosine kinases, which associate themselves with receptors. The ligand that binds to these is called a cytokine, and it can phosphorylate target proteins. This is a diagram of the tyrosine kinase-associated receptors, and these right here are JAK kinase signal transducers, and when the cytokine is not attached to these receptors, the JAKs are inactivated. When the cytokine comes and binds to the receptor, it starts phosphorylation of the tyrosine residue in the cytoplasmic portions of the receptor. And then the STAT protein, which is an SH2 domain, comes to the receptor and receives the phosphate groups and is phosphorylated by the JAKs that have been activated. And then the STAT proteins dissociate themselves from the receptor, and then they bind to one another. And from there, they go into the nucleus, where it binds to the promoter region of the cytokine responsive genes and activates transcription. Another enzyme-linked receptor is receptor-like tyrosine phosphatases, and these work in the opposite way of receptor tyrosine kinases. They work through an intrinsic tyrosine phosphatase that dephosphorylates tyrosines. Receptor-like tyrosine phosphatases are regulated to act at a specific time, so they don't continually counteract what the receptor tyrosine kinases are doing. Examples of receptor-like tyrosine phosphatases are CD45, which helps with the regulation of immune cells and also plays a role in antigen receptor signaling. Another example of receptor-like tyrosine phosphatases is protein tyrosine phosphatase receptor delta. This has been shown to suppress, or it has been shown as a tumor. This example of protein has been shown to work as a tumor suppressor against the childhood cancer neuroblastoma. And they did a study, they observed this by looking at an NYCN amplified cell, and NYCN is also very present in neuroblastoma cells, or neuroblastoma cancers. And so when they looked at it, MYCN belongs to the MYC of the transcription factors that make the proteins that form tissues and organs in an embryonic development. And so it's used a lot for the prognosis, and it's very amplified in higher neuroblastoma. So what they did was they looked at the levels of this protein tyrosine phosphatase receptor delta, and they noticed that there were low levels when the MYCN was amplified than compared to the non-MYCN amplified neuroblastoma tumors. And they also looked in a normal adrenal fetal neuroblast, and they also found that the PT-PRD mRNA expression had higher levels than in the neuroblastoma tumor. From these observations, they gathered that when there were low levels of PT-PRD, that it was more likely a tumor would occur. They also did in vitro and apoptosis assays, and when the PT-PRD protein was overexpressed, the cell lines apoptosis took care of these cells and killed them. So then there was an overproduction of these cells. They also did an analysis of the interaction between PT-PRD and aurora kinase A, and also known as AURKA, and it's overexpressed in many cancers and causes tumors. And it also stabilizes the MYCN. In a neuroblastoma, the PT-PRD dephosphorylates the tyrosine residues in AURKA, which then interferes with the stabilization of MYCN. And if there's not the stabilization of the MYCN, then it's less likely that tumors will arise. Another enzyme-linked receptor are the serine-threonine kinases. These are made up of heterodimers, and there's a type 1 and type 2 receptor. One receptor cannot work without the other. When type 1 is not in complex with type 2, its kinase activity is blocked by the cytoplasmic domain, causing it to remain inactive. The ligands that bind to these receptors are the transforming growth factor and the bone morphogenetic protein. And then when these bind to the receptors, it causes phosphorylation of serine and threonine cells. These also help with the cell growth and differentiation. And this is a diagram of the serine-threonine kinase. You can see that there's the ligand, and it binds to the type 2 receptor, and then this recruits the type 1 receptor, and then phosphorylation occurs for the serine-threonine to go into the cell and perform its functions. And receptor guanylyl cyclases are another enzyme-linked receptor, and this has an unknown ligand, and it catalyzes GTP into cGMP. And the signal molecule, or the way that it does this is the signal molecule binds and begins the production of cyclic GMP, which activates protein kinase, GMP-dependent protein, or PKG, and because of these protein kinases, specific proteins are phosphorylated on the serine-threonine. Receptor guanylyl cyclases are associated with signal molecules of natriuretic peptides, such as atrial natriuretic peptide, and when the blood pressure is high, this hormone is released into the atrium of the heart. The AMP triggers the release of sodium and water from the kidneys, which dilates the blood vessels and lowers the blood pressure. Well, they did research on mice, and what they did with these mice was they inhibited the AMP receptor, and what they found was that these mice had enlarged hearts and their blood pressure rose, so it's important for these guanylyl cyclases to be active and working in cells. The last enzyme-linked receptor that I'll be talking about today is the histidine kinase-associated receptors. These are found in bacteria, fungi, protists, and plant cells and control many of the cellular processes in these cells. They work with a two-component pathway, the input domain located on the outside of the cell, and this is the one that's on the outside of the cell. The signal comes in and binds to it. And then they also work through a transmitter domain that's on the cytoplasmic surface of the cell over here, and this works with the response regulator that will go in and respond to respond accordingly for the function that it's supposed to carry out. An example of this is bacterial chemotaxis, which is the movement of a cell and its environment. And what happens here is that the attractant or the repellent activates a response, and a attractant deactivates a response. If the receptor is being bound by the repellent, repellent binds the receptor, and then it will activate a histidine kinase, an associated histidine kinase, which will then cause phosphorylation, or it'll phosphorylate itself, which is also autophosphorylation, which I mentioned earlier. And then this phosphorylation will go to the regular, or response regular to a protein, and then cause the movement of the cell through its environment. In conclusion, enzyme-linked receptors help to regulate and control an accumulative of cell functions in organisms through their own various mechanisms. And it's very important that we research these receptors, because there are many cancers out there, and if we find a way, if we understand and know the mechanisms, what inhibits and what doesn't inhibit, we can figure out cures to treat specific